Anyways, I was just saying that this is the first week of the course of Dale Purves. So he starts with perception and then moves into neuroanatomy. Studies vision. He's a doctor, MD. Yeah, and so we're going to start by just talking about, what is it that we're detecting? Obviously, we're detecting a small degree of electromagnetic radiation, specifically that from 400 nanometers to 700 nanometers. So we're missing a lot of the spectrum, but however, the idea is that this is what's most relevant to our success, our evolutionary survival, right? Another important thing to note that like the idea of an image doesn't really exist beyond an observer, right? Like an observer is required, a perspective is required to create an image. Reality is just a chaotic bunch of photons and other things for which we as observers can sense, but beyond us, it's really just a chaos of signals for which we can detect. Importantly, there's a discrepancy between, visual perception, or sorry, I'll start with the definition of what is visual perception. Visual perception refers to what it is that we're able to like see and know that we can see it, or can readily see it. So if you think about what you're currently looking at, right, it's like you understand that it's like, okay, I'm seeing something, I'm focusing on it, I'm aware of it, but if I want, I can look at, the color red in the corner of my eye, or whatever, it's all right there. Sensation is more talking about just the sensing of whatever the stimuli is, but not necessarily an awareness or, ready perception of it. But importantly, our perception of the world differs substantially from physical measurements of the world, and in this case, light, right? So if you detect the properties of a photon, it's very different from how you're necessarily going to perceive it, because our perception depends entirely on, the relativity of the situation. And then lastly, there's psychophysical measurements. So we can't, of course, objectively and directly measure subjective experience, but we can comparatively measure it through exquisite or elegant scientific methods. Okay, so let's talk about the discrepancy. So here's an optical illusion. Is there anything here that stands out to anyone, specifically in luminance, how bright an object is? So if you look here, can everyone see my mouse? Yeah?
Yeah.
Okay. So if you look here at tile B is actually equivalently bright as tile A, but you don't see that. In your perception, B seems to be lighter than A, and that's because it's cast under a shadow. So even though the luminance is the exact same in physical objective measurement, these two things are perceived as very different. And of course, this isn't a bug of our perceptual processing. No, this helps us interpret the properties of things in shadows, right? And so this is very important, and it's honestly very amazing that we can do this, but it's certainly not a bug. And if you can't see it, this is one that's not too crazy for me, but if you can't see that these two are the same, try putting your hand across those two tiles blocking it, and you'll see that these things are literally the same color, although you may perceive them initially to be different. Okay, here's one that honestly always amazes me, and I can never quite get it, but I can't rotate it because I don't know how, but like if you were to just draw the dimensions of this table and make a T window, and then you take that window, and it's actually going to fit perfectly on this B. And I know that sounds crazy, but just because of the way the illusion is set up and the surrounding information, you perceive these as being drastically different, but in fact they have the same dimensions. Is that like mind-blowing to anyone? Does anyone think that's just insane, and how is that possible? And Aidan's lying. Here's another one. Dale Purves talks about illusions of angles, and so he shows you a number of 90-degree angles, which are all 90 degrees, but you perceive them as being wildly different. In this example, I'm showing you an illusion of depth, an illusion of length created by these things. So normally, if you learn about these in a class, you don't see this immediately, but what you're shown is this, right? And you may think, oh, well, like, this line is obviously way longer than this line. Anyone see that? This one is longer, correct? Like, you would bet your money that this one's longer. But in fact, these two lines are the same length. And you may again say, oh, maybe this is just a bug in our perceptual processing, but no, we're not directly detecting physical properties of the external world, but rather we're making a sensible model of it so that we can navigate it in a meaningful way. And so what happens is, just because of these angles attached, you perceive this as being longer. And the reasoning is because it's our depth perception. So you see these angles attached to this creates a depth perception that this is farther back than this, where it has angles attached like so. Okay. So if anyone remembers the idea of a controlled illusion from Anil Seth, where he suggests that consciousness and perception is ultimately a controlled illusion, Pervs, he's suggesting that, the illusion doesn't represent bugs, right? They're important biases that allow us to make sense of ambiguous sensory inputs. And importantly, right, like you come into the world with perceptual machinery, sensory machinery to make sense of the world and detect it, but the information is so ambiguous, you need biases to help you interpret it. So here's what I mean by the ambiguity. The ambiguity of sensory information can generally be described by the inverse projection problem. So in vision, this refers to the fact that the retina is a two-dimensional interface to detect the world. But of course, as we all know, there's 3 dimensions of space to detect, right? And so the problem can be defined like this: C equals some relationship between some combination of variables, for example, XA, XB, XC, and they're all interacting with one another to produce C. But if you're given C alone, how can you ever find any one of these or all of these individually? So the example of luminance on the retina, right? Luminance, as it hits the eyeball, so as you get a photon hitting the eyeball, what you're getting is an interaction ultimately between illumination, which refers to the amount of photons leaving a light source, for example, the sun, reflectance, which is the amount of photons actually bouncing off of an object versus being absorbed by the object, and then lastly, transmittance, which is referring to all of the intermediary meddling done by the atmosphere, right? So importantly, the atmosphere is composed of molecules. The air is composed of molecules, and this will distort the light. This will refract the light, so it doesn't all hit you. But yet, we still perceive luminance in a meaningful way, and we can still navigate the lighting of a scene in a meaningful and successful way. This problem, the inverse projection problem, of course, I think I said it applies to other areas of perception. In vision, it also applies to motion. So you can get identical inputs from different kinds of motion on the retina, although they can be vastly different. And that one's hard to believe until you see it. Depth is a really good example. Color as well. We don't detect colors objectively. We detect colors based on the relative wavelengths of things around them. Okay. So to elucidate this inverse projection problem, you can see that this represents the eyeball, right? This represents the lens and cornea, right? And this is the retina. So importantly, the idea here is that these 5 stimuli can all make the exact same imprint on the retina. A, given its depth and orientation, will look exactly like B, will look exactly like C, will look exactly like D, will look exactly like E. And the important thing to know here about the inverse projection problem is obviously this thing can go on forever, right? Like you can just change the orientation of these lines, you can change the size of them, right, and the depth, and you can make an infinite number of stimuli detected on the retina. But somehow, and honestly, it's hard to not call it a miracle or magic. It's like it still can understand what these things are. Of course, you can get illusions and sometimes you're wrong, but for the most part, you're right. And that's honestly something amazing. And, maybe we accredit God or evolution for that. But in any case, it really is an amazing process that this happens. And so, of course, it's important now to talk about what the light is, or what the eye is and what it's doing. So, of course, the main place for refraction is here, the cornea. And so it's going to refract the most light, importantly, get the most light into the eye possible. And then the lens is going to do further supplementary refraction to make it more focused, more resolute, whatever. And yeah, so these will project light onto the retina, where you have all these sensory neurons to detect light and subsequently process it. Importantly, another amazing thing to know, right? So beyond the atmosphere meddling with photons, beyond the properties of whatever the light is hitting and whatever, you also have light traveling through the aqueous humor, the vitreous humor, right? All of these things are also distorting the light. So again, even the light you're physically detecting at the most basic level, it's incredibly distorted. Okay. Oh, and importantly, uh, it's important to note that this is an extension of the brain. This is literally considered a part of the brain. It's the only part of the brain you can always see. Um, and then you have the optic nerve attaching to the retina, where you get sensory information being sent off to the brain or other parts of the brain. And of course, it creates a blind spot, right? So this is blind spot. You can actually find it in your own vision if you close one eyeball and then you look out into space and you move a dot around about 15 degrees away from your focus of whatever you're focused on. You'll find a blind spot where whatever you're holding, some dot or something, it'll disappear. You won't see it. So it's in this sense, it's another case where it's amazing to think that one, our vision works. We literally walk around with a blind spot in each eye, but also it's amazing to think that, like, for me, I'm always just shocked that like we're allowed to drive, like all of us. We're such I don't know, such machinery with such important gaps. I should note that we get around the blind spot by both moving our eyeballs all the time, and also we get around the blind spot by the fact that we have two eyeballs, right? So we have sensory stimulation, sensory input from both eyeballs, compensating for the blind spot in either one. Okay, so importantly, here's a close-up of the retina. So here you have the optic nerve, the optic disc, creating that blind spot, and just about 15 degrees beside it is the fovea. And this is where is the center of your vision. This is where you're fixating your eyeballs every single time. This is what's creating that resolute center of your vision. And then everything else is really responsible for peripheral vision, right? So you can tell there's a very small degree of the eye is actually being dedicated to, that vision that is the most important, arguably. And of course, because the fovea is so important, and you become kind of blind out here, like you really have bad vision. You have bad vision because the word on this page, right? So if you focus on this word, you can detect it, okay? And then if you just stay focused on it, and then you look out, you'll notice that like you can only maybe detect like words in this area, and then beyond that, your peripheral vision can't detect a single word. Like you're kind of becoming blind. And in fact, in the far regions of your peripheral vision, you would never guess this, but you're actually colorblind, right? So you lose all of the cones for detecting color, and instead you just have rods for detecting light itself. So you become blind, colorblind as you move out, but also like your vision just becomes significantly impaired. This is called the macula surrounding the fovea. And interesting fact, 15% of those above 65 have some degree of macular degeneration, contributing to one of the main sources of blindness in the developed world. Okay, and so yeah, we move our fovea around about 1 to 3 times per second, and during that movement, which is called a saccade, you're blind. You're blind during that movement. I believe the saccade, typically travels at a speed of about like, I don't know, 1,500 meters per second, something ridiculous. So you move your eyes very quickly, and for that short period for which you're moving, depending on how far you move the eyes and the orientation, you're blind for about 15 milliseconds, and that happens about 1 to 3 times per second. Again, another form of blindness, but we still compensate and we still successfully perceive the world. So again, it's a sort of miracle to just remind yourself of that. And of course, saccades, interestingly, I didn't know this, I learned this in the course, were first measured by Russian physiologists in the 1950s using a context lens with a mirror on it. And so he would give people context lenses, and he would get them to look at a stimulus, and you could the mirror would reflect onto a photographic paper, so you could get an impression of where was the person looking. If anyone knows, modern eye trackers are very sophisticated, technological, and they look at your eye, and then they basically infer where is your eye looking rather than actually reflecting the light from the eye. Yeah. Okay. And so here's another view of the retina. Importantly, you have these other layers. So again, photons are traveling through these other layers, right? Which are, of course, distorting them, right? So importantly, the ganglion cell layer, the plexiform layer, so on. Of course, distorting the light. And all through these, of course, are blood vessels. Now, the fovea, remember, is the important part for the center of our vision, makes up a small portion of the retina. But these layers dissipate, so that way photons come more directly through to hit the cones and whatever. But still, the rods out here are, detecting very distorted light. So one interesting thing about the retina, here's another image here. So light enters the retina this way, right? So through all of these layers, here's the retina and light's entering. The craziest thing, perhaps, is that, you really do have your photoreceptors at the very back of the eye. And so these are the cones, detecting color, and these are the rods, just detecting black and white light. And of course, it's called a rod because it kind of looks like one, and a cone because it kind of looks like one. And so the reason they're in the back, again, it's crazy because the light now has to travel through ganglion cells, amacrine cells, horizontal cells, bipolar cells. Well, that's actually not crazy, but it's crazy that they're in the back, and again, further distorting the light. But the reason is because they have a high turnover rate for the discs carrying their photopigments, and these discs degenerate, and so they have to be replaced and removed by the retinal pigment epithelium. So that's why they're in the back, interestingly. But it seems like a major design flaw. It seems like something, again, like, the idea of evolution as a tinkerer. It's not a planned engineer. It really does make best out of random solutions. So that's just an amazing thing to reflect on, of how this thing, is responsible for our sight, our vision, perhaps our most important form of perception, and it seems almost deeply flawed. Like any engineer would never come up with a solution like this, but yet it works, and we've compensated with it for millions of years of evolution. So light, directly can enter and stimulate the bipolar cells and the subsequent rods and cones, but also the rods and cones can directly detect light as well. Horizontal and amacrine cells integrate and process information. And again, this is the brain, so it is processing and integrating information in the eyeball itself. And then they're feeding to the ganglion cells, which then go to the optic nerve. Okay. And of course, I think I might have mentioned that as you move away from the fovea, your red, green, blue receptors become less and less common, and you start to become colorblind in your periphery. But in your periphery, you start to get a dominance of rods. Rods become very common, detecting black and white. Interestingly, I remember one of my old professors, she was a perceptual psychologist, and she hated blue Christmas lights. And I found it kind of funny, but she was like, yeah, you can never actually get a good perception of blue and blue Christmas light. And the reason is because you actually have the least cones dedicated to blue, right? And interestingly, you might get some sort of anthropological explanation here, as we're actually very bad at creating language to tell the difference between green and blue, right? So if you look at cultures as they develop language to describe colors, green and blue are something which come after something like red is one of the first colors humans start to talk about, right? So they talk about black, white, and then they add red, and then maybe green or blue. Sometimes they don't even have a difference. So I believe in ancient Greece, they didn't have a difference between green and blue. There are some traditional cultures on the earth today that still don't have a difference between green and blue. And here's an interesting case where language helps us perceive the world. If you show these people a palette with greens and blues, what you find is that if you show them a palette full of blues with one green, they're almost like at random likely to say that the green on the palette differs from the blues, because they're not actually guided by language to tell the difference between the two, right? They just seem like the same kind of color. And so, yeah, this is a very interesting line of work, of course, and maybe some degree of it can be attributed to basic physiology at the level of cones. And so, one brief thing I'll mention is that rods function at scotopic and mesopic luminance. So scotopic vision and mesopic are kind of lower luminance. Scotopic is very low, so you think about getting up in the middle of the night, walking around at night, whatever. One of the things you may note tonight is if you sit in there with the lights off, you're colorblind, right? You're not actually detecting color. Based on your memory and your executive understanding of what is in your room, you may perceive color, but you're not actually detecting it. And you may notice that things become dull in color, and in fact, colors start to change. I can't remember the name of the effect, but our perception of colors change drastically across luminance across the daytime. And so, yeah, mesopic functioning is where you get a degree of luminance for which both rods and cones are functioning. But in normal daylight, you're really just operating under cones with mesopic and photopic vision. And then cones operate up until damage becomes possible, right? So very light, very high luminance for which you're, staring at the sun or whatever. And then, of course, rods determine your absolute threshold for what is it you can see. And so rods ultimately determine what is the lowest level of luminance that you can detect. And that's because rods just operate in a lower level of lighting. And the reason being is because you get about 100 rods attaching to a single ganglion, whereas for cones, you have a 1-to-1 ratio. And so the idea here is that because you have so many rods feeding into a single ganglion in the retina, you're better able to detect a photon by any one of them or any combination of them than feeding to the ganglion in the optic nerve. Whereas for the cones, you have a 1-to-1. So the idea here is you get a greater resolution to detect differences in lighting, but you may not be as great as detecting very low levels of light. Okay, nearly done here. So here's the primary visual pathway leading from the eyeballs to the lateral geniculate nucleus and the primary visual cortex, crossing over the optic chiasm. So one thing to note here is that outside of the primary visual pathway, visual information is being transmitted to other locations. For example, the superior colliculi and the Edinger-Westphal nuclei, and these two deal with unconscious visual processing. So the superior colliculi, they are responsible for orienting the head and the eyes towards just scanning your visual scene, right? So if you think, as you exist, you're not really controlling a lot of your head and eye movements, especially if you're orienting to a loud and salient stimulus. It's really happening automatically for the most part. Your eyes are scanning automatically. Your head is moving automatically without you even realizing, just so you can get a full view of the world. And again, the fovea is so tiny, you have to do this. You have to scan, and you'll miss things because you're only scanning a small portion of the world. And then in higher order vision, you're creating an idea, a model of what exists around you. But again, we really do exist in a model in our head that is not exactly a 1-to-1 replica of reality, which is just an amazing idea to think that you do exist in a continuous controlled illusion. Okay, so an important thing to note about the visual pathway is the contralateral information processing, right? So what you have is that inputs corresponding to the right and the left hemispheres of visual information. So your left visual field is going to be processed in the right side of your brain primarily, and then your right visual field is going to primarily be processed in the left visual field, right? It's kind of weird. Some people ask why, right? One of the hypotheses is just that this is, again, a result of evolution as a tinkerer. This is a sort of, what's the word? It's kind of vestigial, I guess. It's a result of, the idea is that fish have this kind of processing, and it works best for their tail flipper response. So this contralateral organization just creates a better tail flip response, and so this is just left over from that. But yeah, so the optic chiasm is just where they cross over, and then the lateral geniculate nuclei are just these relay centers for visual information. This is another thing for which people just don't understand why it's there. It seems like really unnecessary, right, to send visual information like across the body and whatever, and then you do this and you come down to the primary visual cortex where it starts to get processed. Because what you find is that information leaving the LGN is pretty much the same, right? So and then it starts to change over here, but no one really sees much purpose in this area, possibly like multisensory integration of information, but again, it's a mystery. And so yeah, one important thing though to note about the lateral geniculate nucleus and the primary visual cortex, which is very interesting, is that there's a retinotopic organization throughout the whole thing. And so what you find is that if I stimulate this area of the retina and this area and whatever, what you find is that cells in the LGN, which are dedicated to those spaces, are also organized beside each other, right? So it's retinotopic. So neurons in here are organized relative to their organization in the retina, and this also happens in the primary visual cortex. So within the LGN, you have 6 layers of cells. 3 to 6 layers are parvocellular. The retinotopic, so they correspond with the fovea and what are called peak ganglion cells. So yeah, and then there's 1 to 2 layers are magnocellular, and these refer to the periphery, and they receive their inputs ultimately from the M ganglion cells. So here you have a lot of dedication to fine vision in the fovea and whatever. Here, a lot of processing is dedicated to detecting large changes across the retina. For example, a big moving object, maybe a tiger or something. Okay. Very interesting fact actually about the LGN is that, okay, it's a relay station from the retina to go to the visual cortex, sure. But what you find is that the visual cortex sends information back. And what you find is 10 times more inputs coming from the primary visual cortex onto the lateral geniculate nucleus. And if anyone remembers from my talk of Anil Seth, he heavily emphasizes perception as something which is both bottom up, right, which would be the retina to the LGN to the V1, but also top down. He emphasizes top down as being incredibly important, right? That's our bias in changing how we do bottom up processing, and then you get some mix in between the bottom up and the top down, which ultimately produces what is your perception and consciousness. Okay, so we're almost there. So the primary visual cortex is split by the calcarine sulci right here. And so the bottom half is responsible for binocular processing of information, so information from both eyes. And then the top half is responsible for information process of just one eye, so monocular. In the center of it, what you have is regions dedicated to processing the fovea. It would actually travel down the sulci, right? So it's both monocular and binocular processing, but dedicated to the fovea, right? The center of our vision. And interestingly, what you get, right, obviously, it's kind of obvious, right? So the fovea is a very small part of the retina, but then once you get to the visual cortex, there's a large degree of it dedicated to this small portion of the retina, right? Because that's where the important part of our vision happens, the fine, the resolute area of our vision. So in terms of retinotopy, right, it's retinotopically organized, and things on the retina correspond to one another in their neurons in space, but there is a larger disproportionate amount of area dedicated towards the fovea. I wish I didn't have time to find an image of this, but the retinotopy can be explored by things like modern functional imaging like PET and fMRI. In the course, Pervs cites an example where they trained the monkey to basically stare at this web-looking stimuli. So maybe look at the cerebellum. It kind of looked like that. So it was basically like a web, and the monkey would stare at the web, and it was trained to look at the web, I don't know, for maybe like an hour a day, right? And that was just the training portion. And then they moved on to the experimental portion, or the point where they actually start to image and get a meaningful, experimental outcome. So what they did is they take this monkey who's trained to stare at the web, right? So stare at the center of the web, right? And then, just stare at it. What they did was they injected it with glucose, right? That had a radioactive particle attached, right? And so what you do then is then the monkey stares at it, and it does this a couple days, right? It'll take an hour a day and stare at it. The radioactive glucose will then be sent to areas of the brain processing the stimuli, the web-like stimuli, right? And then you do this a couple times, right? And then they ultimately kill the monkey, and what they find, right, is that you get these radioactive imprints on these parts of the brain, right? And again, I'm just using this as an example of what it would look like, but I'm actually talking about this region. But what they found is that you get this web imprinting of the radioactive chemical overlapped onto the visual cortex, right, with a large degree on the fovea, and then you get the webs of the stimuli itself imprinting onto it, right? And so then they infer the retinotopy, right, because again, the organization of the web stimuli on the retina is maintained in the visual cortex, if that makes sense to anyone. Modern imaging would include fMRI, where you see you're really just detecting what's called a bold response. And so ultimately you're detecting, you know, the amount of blood oxygen traveling to parts of the brain. And the idea here is it's called neurovascular coupling, and you're detecting metabolism, right? So you're just trying to look for what areas are being used. And the idea is that, well, they're more active if they have a higher metabolism than at rest. That's the crucial thing. And then you get an image of what part of the brain is being used. And then, of course, retinotopy is lost in higher visual regions, which are really processing very abstract information. In the primary visual cortex, you're getting things that are very responsive to like simple lines, you know, simple orientations, just lines. The interesting thing, I've heard it explained this way, is not only are they really receptive to lines, very basic stimuli, but also moving things. They really respond well to moving stimuli. And the idea here is that if you're an infant and you're coming into the world with no understanding of what objects belong to which objects, right? Like, you know, I'm looking at a plate and a fork in my own vision right now, and I'm, you know, imagine you're a baby. How do you know that the fork is a part of the plate or not? That's not something you know intuitively. How do I know, you know, maybe is the tree an extension of the ground, or is it a different thing? Well, one of the ways in which we do this is with motion, right? And so we have this perceptual bias in which if two things are moving together, or if a pattern of stimuli are moving together, we assume it makes one object, right? So maybe I take a line and I obfuscate part of the line with a barrier or some bars, but if I see the line moving behind, example, the jail bars, I don't assume there are a bunch of little lines moving in similar space at the same speed. No, I just assume that it's one bar moving behind the blockage. And so the idea here is, okay, so why is the primary visual cortex so responsive to moving lines? Well, because movement of a stimuli creates a natural barrier, and so that way you can detect what object belongs to which object. That's just one hypothesis. Okay. I think we're at the end here. So the last thing I'm then going to talk about, so I talked about retinotopy, right? Retinotopy ultimately invokes receptive field. Receptive field applies to things outside of vision, but in vision, it refers to what neurons are responding to which area of visual field, right? So are they responding to, the area in the center? Are they responding to, I don't know, maybe big areas in the right of the visual field? What? And so what you find about visual receptive fields of neurons in humans is that neurons processing the periphery have very large receptive fields. So if you stimulate areas in the periphery, you're generally going to, if the neuron's dedicated to periphery, it's going to be responsive to a lot of areas of the periphery. But if a neuron is dedicated to, a specific area in the fovea, it becomes very acute. It becomes very resolute, right? So the receptive field declines. And so basically what you get is that if you stimulate a certain area in the fovea, it becomes more and more specific to that area for which the neuron is responding to, if that makes any sense. The ways in which you would typically do this in vision, I believe, is you would use a microelectrode and you would just implant that into the neuron. And so then you could detect from a neuron or a clump of neurons at a very small level, what are they responding to? And then you just show a visual stimuli and you get a graph and you find where is it most responsive to. So importantly, higher order visual areas have very, very large receptive fields, basically the entire thing, because again, they're processing abstractions now. They're not just processing simple sensations and inputs, but now they're processing integrated higher level information, not just lines on the retina or maybe even shapes on the retina, but high abstractions, like maybe trying to identify an object, like is this a chair? Well, now you're not responding to one area of the visual field, but you can respond to any part of it, right? So you're dealing with abstractions at the higher orders of the visual cortex, right? V2, V3, V4, and so on. Yeah, and so that would be it. I couldn't find an image of a receptive field in vision, but I found a quick image of receptive fields for somatosensation, our skin. Is anyone familiar with this here, the homunculus? This is the last slide. I always loved homunculus. I think it's really funny looking, obviously. Does anyone have any idea what it represents? Hello?
Yeah. I don't know. I'm just trying to like think about it, like I don't know, like how it relates to that.
Trying
Like,
to percept the image above it.
Okay, so if anyone remembers what a receptive field is in vision, it's which part of what areas of the retina, what areas of the visual field specifically are the neurons in the brain responding to in vision. Now this is an image of the primary parietal. This is the parietal cortex, parietal lobe. So what you're finding is that if I take an electrode and I stimulate this area, I feel it in my genitals, I feel it in my toes, I feel it in my leg, I feel it in my hip. And what you find is that there's more, these are very few neurons dedicated towards the genitals, the toes, the foot, leg, hip, so on and so on. But you have a lot of neurons over here dedicated towards your hand and face. So like retinotopy, you still have in this other area of the brain, parietal lobe, you have what's called somatotopy, and basically it's like, you know, parts of the hand are stuck together, right? Look at this big chunk of brain dedicated to just different fingers, right? And then it's like, okay, your entire neck, or here, your entire body, the trunk of your body, your leg, versus a single finger, right? So you have a lot of dedication of neurons in the brain dedicated towards the hands and face, and people have made models of it just to better represent. It's like, okay, so the size of homunculus represents how much neurons are dedicated to that area. So this is basically, in other words, kind of your brain's representation of you. It's kind of like how much neurons are each area receptive to each part of your body. And so very little processing occurs for these areas of the body. But hands, for which we touch and manipulate the world, right? These are very important body parts. And then also your lips and tongue are highly sensitive because, again, you're going to be touching world, and the world is going to be entering your body. So it's very important to detect sharp or maybe fine details, something wrong in your food or whatever. I remember one of my professors, you know, he said, I think this is why we probably kiss each other, you know, with the amount of neurons dedicated to the lips and how sensitive they are. But maybe, you know, maybe our kissing of each other resulted in us developing big neural processing for lips, probably bidirectional. Okay.